Record on the cloud. Okay. Okay, we're now cloud recording. Okay, so questions about all of that stuff that I just like ran through at a very fast pace. Nothing? Let me see. Let me get the chat up here. There's no room. Okay, good. Good, good, good. Okay. Yeah, I just had that epiphany of like recording these. And I think because I have like a membership, it's not just the free one, I can upload them to the cloud, or they will be, and then I can just post the link, I think. So we'll try it with this one. We'll see what happens. We'll go from there. And if I could get your help in just reminding me to record when we start, eventually like it'll sink in, but I might forget the first couple times. Okay, thank you. I appreciate that. All right. So no questions so far? Everybody's good? Okay. Excellent. All right. So let us take a look at the syllabus. I'm going to share that with you. Hold on one second. Let me find where I hid it from myself. I think this is it. Maybe. That's not it. Can you guys see this? Can you see something? Sorry, I was switching back and forth. Tammy's dizzy now. Okay. All right, so this is the syllabus. It's kind of the standard. You'll notice that these have been changed to online and or blended while we're in this distance learning format. This is the textbook, as you guys already know, Microbiology for the Surge Tech. Course description is we're going to focus on microorganisms, pathogens, and disease transmission and prevention. Okay, and then a couple important things, like most of this is kind of canned soup, I call it. But a couple things that do change per course are the allocations for the tests, assignments, PPS, and final exam. So that is there for you, and it should correspond to the lesson plan. The grading scale stays the same, of course. It's 77% or better to pass the class. And then these are the course objectives. So if you ever think I've kind of gone off the rails, you can go here and kind of keep me in check with, you know, the things that we're supposed to be learning. So these are them, right? We're going to talk about different microorganisms, talk about classifications, different types of culture medias and fixatives, those kinds of things, what goes on in the lab as far as microbiology is concerned, and what type of equipment. Then we'll talk about morphology of all of those different types of microorganisms, and so on and so forth. And then we'll talk about the different diseases that some of the most common microorganisms, pathogenic organisms cause. And yeah, so I think that's about it. Questions about any of that? I need to get my chat box up here. There's just not enough room for you guys and the chat and everything that I need to have here. Okay. All right, so there's that. I'm going to close it since we kind of went through that. I'm going to now go to, yes, I know it stopped sharing. That's rude, but okay. I'm going to go to the Blackboard, and we'll kind of, the Blackboard is kind of the lesson plan, so we will just look at that. Okay. So if you're looking for lesson plan and syllabus, it's under this course syllabus tab. I realized this morning that I had 2 lesson plans uploaded and not the syllabus and lesson plan, so I did correct that. Make sure that you check your announcements at least once a day because that's the easiest way that I have to communicate with you. So if there's any changes, agenda, whatever the case, I will post it there. Those of you that have not gone to this journal, if you could go there and just acknowledge that you've read those policies, that would be fantastic. And as far as course content goes, you're looking at my view, so yours is a little bit different, but at the top is the link to the Zoom meeting. This is a, the same link will be used for every single course, so you can access it here. I try to post it in the announcement as well, but it's the same link throughout the course. And if you want to get to those course policies, you can also do it from here. And once you click here, you'll just click create entry or something like that, and you can just read and then respond there. So as we kind of get in here, the weeks will pretty much look the same with just a couple differences. So how things will go is from 8 to 9 is kind of typical of what we did in the last class. You'll watch your lecture. First part of this class, we're going to have 2 chapters at a time. And then for the back half of the class, we'll have 1 chapter at a time, and that's because those get a little bit more meaty. So kind of take them in a little bit smaller sized bits. So for every chapter, you'll have flashcards due. And then so today, I kind of went over the content for that and what the homework is. Thursday, kind of the same thing. Shows when your flashcard assignments are due. Reminder that Friday is a holiday. And then when we come back from our holiday, we will have you know, second verse, same as the first. We'll go through chapters 5, 6, 7, 8, 9, 10, 11, 12. And then Friday, guess what we have? Midterm. So put it into perspective, a week from this Friday, you will have your midterm. Okay, it's worth 50 points, I believe. There's a lot of matching in there. Again, terminology is going to be a big deal, so make sure that you study that. Okay, and then each week we have a threaded discussion. For threaded discussions, let's make sure we are putting a substantive initial post. There needs to be research. There needs to be evidence of research. So not only referencing, but also in-text citations. So you'll be graded on that as well as the content of the post. So let's get a good discussion going there. I'm looking forward to reading those and participating, of course. My dog is barking at the trash man. Sorry about that. Okay, so then, oh, let me point out, let's see, where is it? Okay, so Monday of week 2, right here, this is an extra little thing, this first project deliverable due. Okay, so we'll talk about what that looks like. I'm going to pull that up for you in a second, and we'll talk about all the details of the project. So that's when the first part is due. There's 3 parts. Okay, so that one's there. And then business as usual. And then we have our next deliverable isn't due until week 4. And that's right here, week 4, Monday, second project deliverable. And then the last deliverable is going to be due that following day, which is going to be when we begin our project presentations, which I'm really excited about. And I think you guys are going to have fun. And learning should be fun. So just because we're adults doesn't mean that we can't have fun when we learn. At least that's my opinion. So you'll notice that on the back half of the class there, because those chapters are kind of split apart, now your homework is like every other day for those flashcards, and our Kahoots are every other day, post-tests are every other day, so that'll kind of space out a little bit in the back half of the class. And we'll have a little bit more time for those presentations, blah, 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 okay? And then we finish up Monday of week 5. And again, that would just be your survey and your exam and some corrections if you choose to. And then after that, we go into one of your core classes, SUR 140, where we start talking about some basic principles of search text. So that'll be super fun to get back to that as well. So that's the carrot that I'm dangling at the end of this course, is getting into that. Okay. Let's see, let me go over some finer details for you. Any questions so far, since I can't see your faces? You can either shout it out or type it in the chat, whatever you so choose. If you have something, just put it there. I found out how to embed videos. I was all excited about that. So here's your threaded discussion 1. So all you have to do is just click right here and just create a thread. So you are going to write your personal mission statement. So once you click on here and click create thread, you'll be able to see the content. This is just kind of a generic, hey, remember this is what's required. And then in every week will be where you submit your virtual lab. So today you're gonna submit your notes with a couple takeaways, all right? And then I do have the PowerPoints here as well. They don't have any recordings, but they do have interactive stuff attached to them, like videos and stuff like that. So, you know, it's another resource for you to have, okay? So let me just look at one more day because that didn't have homework. So here is your flashcard homework assignment. So it just asks that you use Quizlet and then go ahead and upload that link for me. And it tells me how many terms. It tells you how many terms too. So I'm gonna be looking for 31 plus 24 terms. Okay, that's what's gonna get you full credit. Page numbers aren't required or anything like that, okay? Questions about any of that stuff? Okay. I think that about does it for that. We're going to talk about the course projects. So we're going to look at that a little more closely. Hold on one second. Close your eyes, Tammy. Don't get dizzy. Okay, can you guys see this?
I was talking to my son, sorry. He wanted a popsicle.
He did?
Yeah, I said no after lunch.
That's Ian's favorite is popsicles.
That's probably why I was rolling my eyes.
Okay, can everybody see this experiment presentation document that I have up here? Can you guys see that? No? Oh wait, hold on. Why does it say my viewing is paused? Oh, okay, hold on. I have to stop. For some reason, when you switch, it makes you stop. Okay, let me try again. How about now? Are we okay? Thank you. Thank you. Okay, so this is my labor of love. So I'm excited about this. So what you're going to do is perform some sort of microbiology experiment at home over the next 3-ish weeks, 3.5 weeks. Okay, so this document is attached to every area where there is a deliverable, so you can find it there. Weeks 2 and 4. I believe, weeks 2 and 4. And for each deliverable that's due, you will find this content also replicated there. And like I said, the entire document is attached there as well. If you want to have the whole thing and print it off, and you know, I like to do that and use it as kind of like my checklist. So your first objective for your first deliverable is going to be due on July 6th at the beginning of class. And I have listed some websites here that I found that had some interesting experiments that you could do at home. However, you can find one on your own. I'm perfectly fine with that. If you know of one or you want to do some searching on your own, I just wanted to kind of do some of the footwork for you and give you some resources. So you can use one of these, you can find something else, that's perfectly fine, but you need to be able to do it in 3 weeks. Okay, because, and you're gonna need time to kind of write up your results. So think about the available time that you have to make this happen. And so your first deliverable, and you're gonna write this like an APA paper. So it's gonna kind of be like a lab report a little bit. It's gonna feel like that a little bit if you've ever written a lab report, but it's gonna have a title page. And the title page is gonna have the name of your experiment, your name, date, etc., just like an APA title page would have. And then I try to include some tips in here. So tips for the title page would be that the title should be concise and specific and tell the reader, moi, what you did, okay, or what you're going to do. All right, so that's going to be page 1. So the next page section, you're going to start with the description of your experiment. And you're going to describe the purpose of your experiment. Okay, and the purpose should be stated in 1 to 2 sentences, so it's not going to be a lot, right? The purpose, you don't have to write pages and pages. You should know the purpose of the experiment before you start. Okay, so that's an important component there. So you're going to give a description. Then the next section, which doesn't have to be on a separate page, it can just follow that. Section 2 is going to be your predictions. So before you start your experiment, we actually, when we're doing these classes on ground, we do an experiment with potatoes as a medium, and we try to grow some bacteria on there. Hey, Tammy, you're not muted, VP. Here, I'm just going to mute you, Tammy.
I'll have to see later.
Yeah, you weren't muted. Okay. So as I was saying, the next section is going to be your predictions. So you're going to predict what you expect to find. When I have my students do this potato one in class, I ask them to predict some things like, what types of bacteria do you think you're going to find? Right? And then you have to think about, well, how do I answer that question? Well, we learned about there's some bacteria that like light. There's some that don't. There's some that like starch for food. There's some that don't. There's some that like, you know, carbon dioxide, oxygen, non-oxygen environments. So you can make some predictions about what you're going to expect, okay? And this shouldn't be something that you Googled on the internet to say, hey, what should I expect from this assignment? Like, put some of your own thought and reflection into it and try to tie it into what you're going to be when you grow up. Okay. So that's going to be your predictions part. And then the third section for your first deliverable is going to talk about your methods. So what is your step-by-step? And this can be a bullet pointed list. It doesn't have to be full sentences. You don't have to worry about that. It's just going to be, I'm going to do this, 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 and that. But then you also want to include a list of your supplies. This is going to help you do your prep work for when you actually do your experiment, can help you expedite that process of gathering your things. Okay, so that is the first deliverable that's going to be due on the 6th, which is Monday, right? What questions do you have about that first part? Nothing? Am I going to get a lot of text messages after class? Because I might not respond to those. Okay.
Wait, Miss Windsor.
Yes.
So I'm sorry. I'm just waking up a little bit.
You're good, me too.
So we're writing like an APA paper, or is it more like a, like it's not gonna be like an essay, it's gonna be like a list of all of our stuff?
There's different parts. So
Okay.
this is what is going to be this section. Like when you do when you write up your report, you want to use APA format. So the title page is going to be APA format. And then, you know, you're going to use APA formatting. So 12 font, Times New Roman, double spaced, all of that stuff, right? So that formatting is going to apply. And then when you write your description, you want to use the same thing, right? But then, and when you do your predictions, so that's going to be written like a paper. Potential spam calling me. That's awesome. I don't like spam. And then your methods section, you know, you want to still title it. Title each section and then add your content. Okay, so we're gonna kind of we're gonna apply APA format, but different sections are gonna kind of look different. Does that help?
Okay.
I said no, I don't like spam. Okay, does that help, Alexis?
Yeah, that helps. Thank you.
Okay, good. You're welcome. Any other questions? Any other brave question askers? Please ask now. Be brave.
So we can do, for example, like section 1, 2, and 3, we can do it all in one page, or?
If that's all it takes, sure.
Okay.
Yep. But if you're gonna do your steps are probably gonna be, you know, especially if you're doing double spaced bullets, they're gonna probably kind of draw out a little bit because you want to be really detailed with your steps. Think of it as being as detailed as if you could take that and give it to somebody else and they could do your experiment. Does that make sense?
Yes. So pretty much like a cooking recipe, pretty much.
Yeah, exactly. You're exactly right. That's a good analogy.
So do we have to present this to the class or turn it into, like, do you want pictures with the first deliverable?
That's a great question. Not yet, but that's coming up.
Okay.
So the first section is just going to be this 1 and 2 under first deliverable. Then, and you're gonna, that's gonna be paper-esque, and you're gonna upload that to the Blackboard. Okay, so hold that thought because that is just what we're gonna go into next. So I think I can answer your questions as I talk about the second part and third part. Any other questions out there so far?
So the videos that you have linked right there, they should be able to like help us out to like get an idea of what to do. Okay.
Yep, they're not videos, they're websites.
Websites, my bad.
But yeah, they're websites that have a list of a bunch of different experiments. Some have kits you can order, other things, you know, you can just use stuff that you have at home. Okay. Awesome. Other questions? Now, I haven't written the rubrics for this, but the rubrics are going to follow this information. So if you stick to this information and use it like your checklist, you're going to be golden. All right. So let's move on to the second deliverable, and I'll continue asking, taking questions as you guys have them. Your second deliverable is going to be due July 20th, and you're going to add that to your already existing paper. Okay, so you're gonna go back to the paper that you saved and you're gonna add this content to it. So it's gonna be one fluid document, and then you're gonna upload that to Blackboard on the due date with the added sections. Does this make sense? That's why I put add to previous. That's what that means. So your first deliverable, you're just gonna create your next section, which is your results section, right underneath your methods section. Okay? Rainy, he cracked me up. Okay, so you're just going to add it to your previous paper. The next section is going to be your results section. So here you're going to tell us about the results. Tell me, right? Was there measurements, calculations? You know, this is where any tables, graphs, figures, math, anything like that is going to be contained. And so the instructions specifically say you must have at least one chart, table, or graph. Okay, Tori, you don't have to do math, but you might have to say 25% of the potato that I rubbed on the toilet was covered with schmutz. Okay? So Ian, to answer your question, deliverables 1 and 2 are the same project, but when you submit the second deliverable, you're also going to submit what you submitted the first time, right? Because you're just adding to your report. Does that make sense? So the first one will be just from the first section, and then the second time you upload it will be sections, deliverables 1 and 2 in one document. You're just going to keep adding to it and then submit the second one as your finalized one. This document is attached to where these are due. So Monday, week 2, and Monday, week 4. I believe, Monday, week 4. Okay? Does that help, Ian? Does that answer your question? Okay, you're welcome. All right, perfect. So in your results section, write it out what your results were, and then have at least one chart, graph, table, something fun. Okay, that's going to display your, like if you did the potato experiment and you have like your 5 different potatoes and what you did with them, you could make a table and have that listed, and then how much growth was on each one, something like that. And you know, it doesn't have to be fancy schmancy, but it has to have one of those. And then it has to contain at least 3 photographs of your experiment. So you can do, my example was minimum of 1 before, during, and after, or whatever. I mean, it doesn't have to be that way, but I think that kind of makes sense. If something else makes sense to you guys, that's fine. If you want to have more than 3, that is fine as well. What else was I gonna say? Oh, if you want to do a like a video of your experiment, you can, you know, post those, you could add those links, like here's my before, here's video of during, here's video after, I don't know, whatever you want to do, photos, video, you can. Okay, so I just thought photos might be easier. I wasn't sure at first. I was thinking video would be really awesome, but you guys can do whatever you want there. So some tips for your results, and you guys can read through these. I'm not going to read through these, but it talks about measurements, calculations, tables, what that formatting would look like. Okay. If by chance, this one right here, V, plate counts, if you're going to, you can buy like little Petri dishes or agar plates, and you can make your own agar. If you're going to do something like that to grow bacteria, then you might want to say, you know, 3/4 of the plate was covered. I mean, we're not going to be able to count them, right? So you could just give an estimate there. All right, and then this kind of tells you about the formatting. Not going to be too persnickety about that, but if you give me a table, tell me what the table is. I don't just want a graph dropped in the middle of your paper and I'm like, okay, I don't even know what this is. So title it, give me an explanation, something, help me out. So then the next section, section 5, is going to be your discussion and your conclusions. Okay, so what observations did you have? Did you see? And what do your results mean? Okay, and so I give some tips there. Discussion interprets the meaning of the results, draws conclusions. And then something else that is important to address in that discussion area is discrepancies. So let's say, for example, I'm going back to the potato experiment, that my control potato that was supposed to have no growth had a lot of growth. More than the one I rubbed on the toilet, and more than me and my 6 brothers and sisters passed around and touched the potato. Okay, that wasn't expected. It was expected that the others would have more growth because people touched them and people are, you know, using the toilet, but not the control, right? So you might want to discuss in this section then, like, why? Like, why did the control have all this growth? And then speculate why. Maybe I had poor technique, right? When I was putting the you know, cut potato that was just going to be the control, maybe it got contaminated or something of the sort. So you want to speculate there. If you do have discrepancies, then why you had them, okay? And then last is just any implications. The meaning of your results, summarize in a couple sentences. Okay, you know, and here we're not really in a real lab where we're like, you know, our research is going to mean a lot and apply to future studies or whatever. But here again, you can kind of speculate and reflect and say, well, this is the determination. You know, the potato that had multi-hand contact had the most growth. And that really means that there's a link between hand washing and, you know, the spread of disease. And so, you know, we should wash our hands or whatever the case may be. All right. And then the last part is your reference page. So you want to make sure that it is titled references. I don't want to see works cited. That's not APA format. So if you have questions, you can go to Al Purdue right there. I put the link for you. Okay. So have a reference page. And then you will turn that entire paper, all of those sections, deliverable 1, deliverable 2, you'll upload all of that to the Blackboard. Questions? No questions on all of that? Are you guys still out there? Because I can't see your faces, so.
Still here. Figured
Okay.
out how to properly work the mute button. Sorry, so sorry.
You're good. You're good. Okay, good. Thank you. I appreciate it that you're, at least I'm not alone in this. This whole page is on the Blackboard. It's there twice. Okay. My suggestion is print it out and use it as your checklist. Okay. I tried to make it detailed so that it would be clear. So hopefully it is. But of course, as you stumble across things, let me know. I tried not to put too much detail in here as to kind of pigeonhole you. I want you to kind of put your own spin on it. It has some autonomy. So I'm excited to see what you guys come up with, what you do. I really loved your portfolio projects. Those of you that did videos was awesome. You know, there was a lot of awesome diagrams and models and those kinds of things. And so it was really fun. I just wanted to tell you guys, looking at all of your projects. So I thought, you know, we could kind of do something similar here. All right, that's all of my blabbing for that. And then the third deliverable does not have anything to do with your paper. You've already turned that in, but it now is going to be your presentation. So you might want to work on them simultaneously as you're kind of writing your report. And then there should be a minimum of 8 slides. You probably don't want to have too many more than that, because 10 to 15 minutes is going to be your max. All right, so at 15 minutes, we're going to draw the line and say, wherever you're at, I'm sorry, we have to move on to somebody else. So, you know, condense it, just put the really pertinent stuff in there. We like to see pictures. If you made a video, we might want to see a little snippet of that. You know, canned soup, just give us the exciting stuff. Okay, you don't really have to go through a lengthy step-by-step. Okay, brief overview of methods. Slide 4, brief overview. All right. So this you will upload to the Blackboard as well, but then also be prepared to share it when we start our presentations. And we're probably, I have an app on my phone that just randomly like chooses individuals. So I'll probably just create a random list and then we'll just start out. And as we have time through the rest of week 4, we'll go ahead and do those presentations. Okay. That's it. In a nutshell. Yeah, it's going to be fun. Once you get to sit down and look at this, I think hopefully you'll get excited. All right. Okay. Anything else about this? Let me show you where in Blackboard it is located really quickly. Okay, here we are back in Blackboard. The first deliverable is due week 2 on Monday. So if you go there, and here is the attachment right here, week 2 Monday, first deliverable. There's all the stuff we talked about, but the entire document is here. Okay, and then let's just go to where the second deliverable is due, which is week 4 on Monday. And here you'll see the second deliverable. Here's all the spiel, which it didn't really format very well, but it is all there. And here's the document again for you. Okay, this is your entire report where you're going to upload that at. And then week 4, Tuesday, comes on a Tuesday, is the document a third time and third deliverable with the spiel. And I did put on there 10 to 15 minute maximum because we have about 4 hours to do these presentations. And at 10 minutes, maybe I figured it at 15 minutes, it's gonna take us like 3.5 hours. All right, so we do have a tiny bit of wiggle room, but anyways, so that's where you can find it, okay? 3 places. All right. Ta-da, done. Done with that part. Okay, we did it. Where's my chat box? Oh, here it is, okay. Anything else? Any other questions about that before we move forward? I see it in week 4, the third part, but I don't see the other parts. Okay, so if you look in the Blackboard on the weekly content, wherever it says it's due, that's where you're going to find it. Okay, so deliverable 1 is in week 2 on Monday. Deliverable 2 is in week 4 on Monday. Deliverable 3 is in week 4 on Tuesday. Okay, that's where you'll find those. And you can also look at the lesson plan. You can also look at the Blackboard. It is your lesson plan. So if you do all of that and you still can't find it, then let me know, okay?
I did go into week 2 and clicked Monday, and it just shows flashcards, Robert Koch, and microbiology, chapter 5 and 6.
Same for me.
Okay. All right, stand by. Let me see. Let me see why. So that's week 2 where you're not being able to see it, correct? Let me see if By the time I did this, I think my eyes were getting kind of crossed. So there's so many details. Maybe I clicked. Oh, yeah, I see the problem.
I see it in week 4.
Okay, now you should be able to see it. For some reason, I must have accidentally checked to hide it. But it should be available now. Somebody confirm or deny. Okay, thank you for letting me know. I appreciate that. Okay, that's why like your view is different from my view, so. Okay, yeah, I didn't want to scare you. I think because there was an old project in there, I think I know why now. And I hid it because I didn't want you guys that were like, you know, early birds to start working on a project that you weren't going to be doing. So I hid it until I could change the content and probably just forgot to unhide. I didn't build that one. I built the other ones. I just used the old project. So you can see all of them now. We're good. Okay, thank you. I appreciate that. All right. Okay, anything else about that? Okay. All right, so do you guys want to take a quick break before we start talking about chapters 1 and 2? Okay, so it looks like it's about 10 till, so why don't we meet at the top of the hour and we'll pick up with chapters 1 and 2. Okay? All right, I will see you in about 10 minutes. Okay, we should be back recording. All right. So here is the plan. The plan is I'm going to break you into groups and I'm going to assign each group with a question, except for one group has 2 questions, but one is really easy. So you'll each get a question, you'll go to your groups. I'll give you about 10 or 15 minutes to hash it out and get an answer. And then we'll come back together and we'll have a discussion. Okay, first though, I skipped my why do we care? So I just want to talk a minute about why do we care? I got my whiteboard. I don't know if this is going to work or not, but we'll see. If it doesn't, we'll never do it again. I can bring this closer so that you can see. Can you guys see? Maybe. Okay, we'll see what happens. So who can tell me why microbiology matters to us as surge techs? Shout it out.
We need to keep the room sterile to prevent infections.
Sterility. Awesome. Can you see it? Kind of? I don't know. Okay, what else?
It's one of the bases for aseptic technique.
Yes. That's important to us, right? It's our rules of engagement. What else?
Patient safety.
Patient safety. Good. What else?
Understanding microbes leads to understanding what to do about them.
Okay, so if we know how they work, then we know what to do.
We
Kind of
also
along with that, piggybacking on that a little bit, just understanding how they spread also.
Yep, I like it.
And to go along with Tammy, not only patient safety, but our safety as well, like we're seeing with all the medical professionals right now with COVID.
Personal safety. That's a big one, right? Because if we can't keep ourselves safe, then how can we keep our patients safe? Right? So that's an important one for sure. Come over here on the side. Okay, what else?
Well, the safety of our teammates, you know, you mentioned that, like, if we see that the physician's cap or whatever touches a non-sterile area or is out of that zone, then we need to say something. So we've got our patient safety, we've got our personal safety, and then we also have the safety of our teammates.
Safety of team. Indeed. What else?
PEP and PPE.
PPEs?
Yeah.
What kind of PPEs? What do we have to wear as surge techs?
Gloves, masks, gowns, goggles.
And like we just talked about in these two chapters, if we know that there's a specific microorganism that may be present, then we need to accommodate for that as well.
Right, like the HEPA mask, right? Because if we have like TB or coronavirus, then we're gonna have to wear some sort of mask that is different than just our traditional one, right? Yeah. Yep. So isolation precautions, maybe we should write that. Or transmission-based precautions, I think the book calls it. Transmission-based precautions. Anything else?
Post-exposure prophylaxis, like knowing when to take it if we were exposed to something.
Yes, indeed. I actually had a student that she came to campus, I don't know, for whatever reason, and she was telling me that she got stuck, and I'm like, oh my God, you got stuck like with a dirty needle in surgery? Because she was on her clinicals, and she's like, no, I was actually at the park with my daughter, and we were sitting on a park bench, and there was a dirty needle that was like laying in between the slats, and she went to scoot over, and so she moved her hand like across the bench, and she felt a stick, and it was a dirty needle. So I'm like, you need to go, like the whole prophylaxis, you know, lecture. And so she did, and she was fine. She was okay, but that's just super scary, right? The thing about that makes it even more scary is we have no idea who it is. But in surgery, we do know who it is, and there is a chance that if that patient consents, you know, we can do a blood draw, we can test their blood, so that can help us out. But yeah, for sure. Prophylaxis. What else?
Along with patient safety, we use it to prevent surgical site infections.
Ah, good one. Prevent SSI. You only have to fuse one person's knee joint from an SSI, and it's all very clear at that point.
I was going to say, I agree with what chapter 1 brings up with understanding the history of microbiology helps us understand where we've been and where we are going with it. Like, that makes sense.
For sure.
Like, I was pretty blown away that abiogenesis was still around in the 1800s. I thought that was ridiculous, but knowing that, it makes more sense.
Yes, that's so crazy. And that, you know, what blows my mind too is that there were these little bits, you know, of proof, quote unquote, or something that should have, you know, provided a bit of doubt as to spontaneous generation of life. But you know, experts just refuse to believe it. Like, oh yeah, no.
Well, and the time frame, you know, like Ian is saying, the time frame from, what was it, the 1500s, where it kind of got brought to light a little bit and everything, and it took until the 1800s. The time gap is fascinating that it got, you know, pushed off for so long.
Yeah, and even in the mid 1900s with puerperal fever, you know, and Semmelweis saying, hey, you know, I've observed that midwives are washing their hands in between patients. I think that we should, you know, and they just kind of, what would my grandma say, poo-pooed them, like, yeah, no. And
burn them, burn the witch.
Yep, exactly. And that was, you know, really not that long ago. That was.
It's like,
yeah, and then like once
extra work.
Right.
Sorry, Ian, go ahead.
No, I was just gonna say, and then like going into the 1900s, once it was accepted more readily, like the science in front of them, once that happened, I think they called it the golden age of microbiology. Like once it started, it just kept going, and you had this period of like 1500 to late 1800s where there was some development, but not much. And then once it was finally taken seriously, it was just, let's get on the ball and figure out everything, and which, you know, we're not even close to, but that's impressive. 100 years we came farther than we did in 400.
Yes, for sure. Yep. Awesome. Good discussion, guys. Thank you very much. I appreciate that. Any other thoughts about why we care? I think this is a really good list.
The only other thing that I can think of that I take into consideration is the immunocompromised patients. You know, what do they already have? What do we need to take extra precautions with? And just because they are a little bit more sensitive to certain things.
Yeah, immunocompromised. Yeah, for sure. That's when our sterile technique is even more critical. I mean, it's critical across the board, right? But you know, that's why with like total joint replacements, they put these big signs on the door that says no traffic. Like they don't want in and out, in and out, in and out when they're replacing somebody's joint. You know, in the situation that I've been in, we had a patient come back with a infection. Now, they didn't specifically point fingers and say it was a surgical site infection, but the knee was infected. And when you have a total joint that's infected, you take it out. So the implants that we glue in with cement now have to be hammered and chiseled out of the patient, which is going to remove a lot of bone. And then guess what? They get sewn up without a knee. So we take a cement and we mix it up and we put a whole bunch of powdered antibiotics in it and we make a big block out of it and we put it where their knee was between their tibia and their femur and we sew them up and we send them home, right? And then when their infection is gone, hopefully, and their white count looks normal, we bring them back in, we clean it out, and we put in a new knee. So, but now we have to do a revision and there's a lot less bone. And so it's like Legos to build this person a new knee. And this particular situation, he developed a subsequent infection, which they think his primary one just wasn't gone yet. And so then we brought him in a third time, took out that knee and fused his joint. So something called a Wichita nail. So now he has no joint, he can never bend, you know, never have the ability to bend his knee again. So, you know, those are critical things for sure. It's scary. It really is. You know, the goal is, I'm not saying that I don't care that you know the difference between a hemostat and a Mayo-Peon when you go out to clinicals, like you will, but if I had to choose between that and sterile technique, like sterile technique is what you have to have developed such an understanding and just muscle memory of sterile technique that by the time you get out there, you don't even have to think about it. right? By the time you get out there, you don't have time to think, is this sterile or is it not sterile? Was that contaminated or was it not contaminated? You need to be to a point where beyond a shadow of a doubt, you can recognize contaminations, you know how to fix them, and you know how to stay sterile. Those are the most important things in my mind, because that's what's gonna, you know, hurt our patient the most. So good. Any other comments about that? Move this back.
So follow up on that. I know, like when surge techs bring in the case cards to decontamination, you know, most of the time we can't even see the instrument used or touched, but a lot of it does have to do with not being able to see the microorganism, but you still have to use the right chemicals in that procedure. Knowing the transmission or the category of what it is, you use different chemicals, which is safer for everybody around us.
Yes, absolutely. We're going to talk about that a little bit more, Ruan, and that just makes me think how extremely important sterile processing people are. You know, because we take certain measures to verify that something's been sterilized, but when it gets into the room, we can't look at an instrument unless it has bio burden on it, unless there's a chunk of bone stuck in it somewhere, or, you know, blood that we can physically see, or tissue. We have to assume that there's no microorganisms there, right? And so that's where sterile processing's job is so important, and it's so integrated with what we do, where we're going to talk about how what we do in the operating room is directly related to what sterile processing does, and what we can do as surge techs to start thinking forward to that next process, you know, that next step in the whole process. So yep, absolutely, Ruan. Anything else?
And there's a big controversy right now. I mean, debating if N95 masks could be re-sterilized, or I mean, right now we're just marking them off for 10 times. It's used 10 times, and then you toss it. And where do you toss it, and how does it get, you know, out of the OR?
Right. Absolutely. Yeah. Then, and you know, the book talks about how we have these, what does it say exactly? Heavily dependent on scientific research, right? So in my mind, that makes me think of evidence-based practices. And evidence-based practices are practices that we implement because research shows that we should, you know. And so right now we're in some new territory where we're having to adapt and evolve our practices, right, to a new situation. So and there's you know, some growing pains, if you will, with that.
Well, and not only that, but then we have these pathogens and viruses that like to mutate.
Yeah.
And change. You know, we've got resistance to certain antibiotics, and so we're having to step up our game. You know, it's the same virus, it's the same genetics, but these little suckers, you know, they're smart, and it's survival. So that's exactly what they're doing as well, which makes our job even that much more imperative. And like the gentleman was saying, you know, and the instruments and things like that, you know, what's the process like? How do we have to sterilize them? How do we handle them if these things are just becoming more resilient?
Absolutely. Absolutely. You know, there's a lot of organisms that our antibiotics can't kill now. So there is a chance that, you know, if a surgical site infection is caused by MRSA, then we're not going to be able to cure that patient. So 1 in 3 people are carriers of MRSA. So there's a few of us in here that are carriers, statistically speaking. So yeah, good discussions. Good, good, good. I want to continue this, but I want you guys to continue it in your groups. So right now I'm going to attempt to put you into some breakout rooms. And I'm going to choose 4 rooms. See what that looks like. Okay. Yay, I actually did it right this time. I don't have to reassign. Okay, so group 1, I'm going to call out your names, and if one of you would volunteer to be the group leader, that means you have to have a computer so that you can share your screen or whatever if you need to. So Alyssa, Gabe, Ivana, Jonathan, Robbie, and Wendy. Who wants to volunteer to be my leader in there? Thank you, Alyssa. I think you volunteered last time, my dear. Appreciate it. All right, group 2, we have Ariana, Hannah, Jacob, Rainey, Rulan, and Tallman. Who is game in that group? Thank you, Rainey. I think you were volunteered last time. Thank you. Okay, number 3, we have Alexis, Ian, Jackie, Lixie, and Tammy.
I don't mind. I'll volunteer. I don't really know what I'm doing, but I will.
You're good. Okay. And then my last group is the rest of you, hopefully. Adriana, Andrea, Sarah, Sean, and Tori. Who is up for it? Thank you, Adriana. I appreciate it. Okay, so one last little tidbit of information. Make sure you guys have your textbooks. And we're going to look at some of the under the microscope questions. So let's see, let me turn to the first ones. Group 1. Group 1, you are going to do chapter 1 under the microscope, number 1. So it's on page 16 under the microscope for chapter 1. And group 1, you have number 1. And group 2, you have number 3. Okay, page 16, group 1, number 1, group 2, number 3. Okay, now we're going to move to chapter 2, and that's on page 25. And group 3, you are going to do 2 questions just because I need someone to identify what the disease is. So you're going to do questions number 2 and number 4. So number 4 is just basically saying this is what the disease is. And then number 2. And then group 4, you're going to get number 3 in chapter 2. Okay, questions about this before I send you off to your prospective breakout rooms?
So with this, do you want us, because I know we're taking notes for class, do you want like the leader to comprise the notes for like the own thing in the group and then just present like we did last time? I know some people weren't in that class.
Yes, thank you. So for this, it's just like our class stuff, not our lab stuff yet. Okay. So it's not going to be required to be uploaded. So you know, you can take whatever notes you want or not take notes, that's fine. But when we talk about our first principles and practices, that's when you'll need to take notes each individually because that's what'll be uploaded. All right. Okay. So, but still, you know, once you guys get done, if you want to pick a spokesperson, and then we'll kind of go from there. Does that help?
Yeah, thank you.
Okay. Awesome. All right, so I'll be jumping around, just popping in, checking on you guys in your rooms, making sure that you're doing okay, and that you don't have any questions. So I probably won't say anything, but if you see me in there and you have a question, just shout it out. And then when we have 1 minute left, I will go ahead and post an announcement, and then we'll bring you all back to class. Okay, so I'm gonna give you about 10 minutes, and then I'll see where we're at if we need another 5, okay? All right. Here we go. I'm shipping you off. Okay. All right. Well, we are going to jump next to Rainey's group, group 2, and they had number 3 under the microscope question. Are you going to talk about that, Rainey?
Yeah, I can. Jacob took notes, and I just have to figure out how to get to that.
I can talk about it if it's easier for you.
Oh, that would be great. Do you mind?
Yeah, go ahead. I'll go ahead.
Oh, cool.
So the question is, again, which vaccinations are required for personnel in the operating room and why? We had a couple. We had the hepatitis B, the meningococcal, which is every 5 years, the Tdap, which is every 10 years. Some require the influenza, which is only valid for a year. Varicella, which is the chickenpox, the MMR, measles, mumps, and rubella. And to a why is physicians and nurses take care of the disease and preventions for all patients, so they're constantly being exposed to harmful diseases, and they're a key component to be safe in the OR.
Yes. The only one, and it's really not a vaccination per se, but is the TB test. And that's an annual thing like the flu is. Typically 2 MMRs. Sometimes if it's been a really long time, they might suggest that you get a booster, but if you can prove that you have 2 MMRs from like when you're a kiddo, you're good. Hep B is a 2 or 3 shot series, and it takes a while. So these are all the things that we require for the program as well. And then the flu is pretty much mandatory now. And clinical sites are saying if you don't get the flu, like there used to be a declination where you could say, yeah, I'm signing this thing that says I don't want to get it. There was a time when there was this shift to where they said to their employees, if you don't get the flu vaccine, then you're fired. So we suggest that you get it. So it's pretty much a requirement across the board for you as clinical students as well. So, yeah.
Well, here's another question for you that I didn't see, but, you know, I mean, you're dealing with elderly, you have whatever, and I'm sure that you've got some more senior people working in the facilities. Do they have to get, like the pneumovax, do they have to get the, oh my God, why can't I come up with it? What is it called when you get, it's not rabies. What is it called when you get like, it's like from the varicella and you're old and it follows the nerve line. Yes.
Shingles.
There we go.
As far as I know, those things aren't mandatory. You know, this list here, you know, that Jacob gave that you guys came up with is definitely mandatory, but I haven't heard of those being mandatory at all. Yeah. Good question. Any other questions? Awesome. Okay, yes, get your vaccinations if you haven't got them already. It will not be long before you guys will be stepping out into the field. Excellent. Okay, so now we're going to shift over to chapter 2, and my group is group 3, Tammy's group. 2 and 4 were your questions.
So for ours, the first thing, do you want me to go in order by questions, or does it matter to you?
You can say what the disease is maybe, and then circle back. That might make more sense.
That's kind of how it made sense to us as well.
Yeah.
We decided that this individual has CJD.
Okay.
Due to the description and with the dementia onset, as well as they had lived in England approximately 10 years ago, so there was that exposure there. So we made that connection. And then we decided that to answer question 2, why would this particular disease and surgical procedure be of special concern for the handling, decontamination, or processing of the instrumentation? Well, with as the discussion that we talked about in class, this is certainly something, it's a prion, and it's very, very resilient, very difficult to kill. So there are extra precautions that need to happen. It's something that is found in the brain or the nervous system tissue, and there would be exposure due to the craniotomy for biopsy. And so it has very lethal proteins, and I'll let, I think it was Alexis and Ian actually looked on the CDC website to find some additional sterilization techniques in addition to what we already do.
Awesome. Sounds
So
great.
if you have anything to say about that, they're more than welcome.
So the CDC, it recommends that you immerse like any instruments that you can in a sodium hypochlorite solution for like an hour, and then it gets sent off to routine sterilization. And then it said like some instruments like they'd be damaged by the sodium hypochlorite, so like they'd use like heat procedures to kill any of the CJD. And then it said like disposal of like tissues and stuff like that, it should be like incinerated.
Scary stuff. Hard to kill. Boo. Okay, very good. Questions, comments about that?
So would disposable instruments be an option in those kind of cases, like as many as possible, just to, I don't know, kind of eliminate the need to do some of those sterilization techniques?
Yeah, and that's a good segue into our last group. They had the task of looking at the AST website and seeing what AST suggests, but there really is, and maybe Rulan, I don't know if you can speak to this too, a fail-safe way for killing prions. But you know, the more stuff that we can throw away, definitely the better. Yeah.
Yeah, I was gonna say, I mean, I was on the HM conference for this year because it was canceled, and that's what they focused on. Everything's just thrown away, and then certain disposable, just because it's so high and transmitting.
Yes, yes, that's what I've been finding too. There isn't a sterilization method that we have yet that can kill it. Because the methods that we have work by like killing the processes that go on in the nucleus, right? By denaturing the processes of like translation, transcription, all of that stuff. So because it's just a protein thing. So it doesn't have any DNA. Yes, ma'am, Tammy.
So basically, any other, I don't know if this is correct to say, but any other microorganisms are able to be denatured, except prions are not able to be denatured, and that's what makes them so resilient. Okay.
Correct, because there's no RNA or DNA material. There's no nucleic material.
Okay.
Yeah, good question.
That's scary.
I know, it's horrifying. And what if you dig around in someone's brain and you don't know that they have CJD? I mean, you know, it's like latent for 20 years. So, you know, it's, yeah, I've dug around in a lot of people's brains. Who knows? I dig around in mine most, though, figuratively. All right, good job, excellent. Let's finish up with group 4. Adriana, they had number 3, the AST guidelines. What did you guys find out?
Okay, so one of the most important things they talked about is during the procedure to make sure that your instruments stay moist. You don't want the decontaminated instruments to dry up, and then it's even harder to clean them after that. And then it also said that with instruments that have been in contact with high-risk patients or like high-risk tissue, you want to decontaminate those right away. The surfaces should be covered in material that can be destroyed as well. And you don't want to use splash sterilization with instruments that have been contaminated with high-risk tissue. And then the facility will also have a tracking system for devices that were used with high-risk patients. It'll say like the patient's name, the surgeon, the date that they were used, and just keep track of them. And then also if the patient hasn't been diagnosed, if they're still waiting to know if they are high risk, then you should just quarantine the instruments before they've been destroyed to minimize like the amount of instruments that are destroyed if they don't need to be. Yeah, that's about all the notes that I have.
Awesome. So AST is a really good resource for us to just know what we're supposed to do, how we're supposed to handle situations. So it's always a really good place to look if you have questions, you know, or need guidance about those kinds of things. Awesome. So any other questions, comments about these, this material, the material in chapters 1 and 2? Anything kind of lingering out there? Nothing? Okay. Well, do you guys need another break? Are you okay for another half an hour? You want take like 5 minutes? Okay. I have 10:52, so if you want to come back just a hair before 11, and then we'll pick up there. Okay? Awesome. I'll see you guys in a few. All right, so to finish up here, we're going to start talking about sterile principle 1. And there's a whole bunch of bites and bits to principle 1. So we're going to look at the first 3 parts of sterile principle 1. I have a couple videos for us that are like 3 minutes long, each kind of having to do with these little principles that we're going to talk about. So I'm going to share my screen with you right now. And we're going to look. And I can also upload this to the Blackboard if that would be helpful for you all. So I'll go ahead and do that after class. Let me just write myself a note because I didn't do that.
And this, Ms. Windsor, is for our lab activity, right? We can start this now.
Yes, ma'am. So go ahead and get out something to write with and something to write on. This is what you will upload for your lab activity. Yes, thank you so much for helping me remember to say that. Okay, so here we are at principle 1, and principle 1 is this: a sterile field is created for each surgical procedure. All right, so we don't use one sterile field for all the same patients, and we know that, right? So I'll just kind of read the narrative here, and then we'll talk a little bit more about it. We'll watch a couple videos. So principle 1, the primary method through which microbes are kept to an irreducible minimum in the OR is through the creation of a sterile field for each procedure. The sterile field is a separate sterile area that in a larger sense consists of the surgical site itself. So We start at what's called the back table, and you can see it here in this image, and this is the little ring stand over here. So this is where we start setting up the sterile field, always. We always start with opening a cover on our back table first. That's an important takeaway. Okay, so this is where we're at. Now, once the team is in and they're all dressed and ready to go, and we put those sterile drapes on the patient, then the patient becomes the center of the sterile field, and we move all of our sterile stuff up around the patient. But while we're setting everything up pre-operatively, we always start at the back table when we start opening our sterile things. Okay. Let's see, the drape portions of the patient and the OR table, the sterile portions of the gown and gloves of the sterile team members. So we'll talk about what all those parameters look like, and the draped ring stand that you saw. The Mayo stand, which is the tray that goes over the field, and the back table. So all of those things are going to be considered. Within the field, we have all of our to do that surgery. Okay, so that's kind of the overall. Now, as far as our first principle, let me get it a little bit bigger here so that we can see it. So the first thing is the sterile field should be created as close as possible to the time of use. That's an important thing. We don't want to, if we have a case scheduled at noon, we don't want to set it up at 7 or 8 when we first come in the morning. We want to wait till as close as possible to that surgery start time. Why do you guys think that is? Any ideas?
So no cooties can make their way in and land on them and grow on them and then end up being introduced into our patients.
Yes, we want to limit the number of cooties. You're absolutely right. So the thought is that the longer sterile stuff is exposed to the environment, the higher the probability is that it has become contaminated, right? And so the second part of that is that it needs to be monitored at all times. So it's, you know, our sterile stuff is like the little baby ducks, and we're the mama duck or the daddy duck, right? Once you open something sterile in the room, somebody needs to be in there at all times to monitor it. Why? Do you think stuff is going to jump off the table and crawl away? Why do you think that's the case?
Well, accidents can occur. Anything could be touched and be breached. So, you know, someone monitoring can avoid any accidents like that.
Yes, you're absolutely right. And can flies get in? What do you guys think?
That's impossible.
It's possible. I've seen a fly in the OR before. During surgery, one landed on my Mayo stand. During surgery. That sucks. You don't want that. So monitoring it at all times is hopefully going to help us prevent those things from happening or catch those things when they do happen. Okay.
What's the protocol for when a fly does get in there?
So it depends on the when. If, let's say, I've just opened everything up on this back table, the patient's not in there yet, and a fly lands on it, I'm probably going to scrap the whole thing and go get new stuff. That's the best thing to do. However, if you're in surgery and you have someone open from stem to stern and a fly lands on your back table, you're not going to scrap the whole thing and start over. So at that point, it's damage control, right? Where did it land? Okay, I'm going to isolate that area, isolate that stuff, not touch it, cover it up, whatever is safe. And if I need to get additional stuff, then ask my nurse to get that stuff for me. And kill the fly. Not us, but somebody that's not sterile. The nurse becomes the fly hunter. If that makes sense.
Like, you know, it's not easy to kill flies. That's what I was asking. So.
Yeah, you know what they usually do, and I don't know if this is like falling out of vogue because we don't have hard copy x-rays anymore, but most operating rooms have those like view boxes, you know, the light boxes where you put the x-ray up. And now that we have digital x-rays, I guess you could use the computer screen, but what we used to do is turn off all the lights except for the view box, and the fly will go to the light and then smack.
How exciting.
Yeah, but surgery usually stops, right? If there is a fly, we're gonna stop, and you know, we're gonna pay close attention to things to make sure that we can get that taken care of. So yeah, it's like a big to-do. Good questions. Okay, so that's the first one. Sterile field should be created as close as possible to the time of use and be monitored at all times. Okay, what if a case is delayed? Does somebody have to sit in there and babysit it? What do you guys think?
Can we cover it?
Good question. You are
Depending on how much time.
You are so good, because look at what the next one says. I think it's the next one. Look right here. The sterile field should never be covered with a sterile drape. This is talking about moving it. But the reason why we don't want to cover it is because it's almost impossible to get that cover off without contaminating your stuff. And the reason is because, and we'll talk about the parameters of things and where it's sterile and where it's not sterile, but where a table cover, let's say, think of it as like a tablecloth, right? If you put a tablecloth on your kitchen table, the top of it, if it were a sterile table, would be sterile, but nowhere else is sterile. So where it falls off the edge is no longer considered sterile. So trying to get that cover off because it has been draped over the edges is gonna have to come up and over your field, right? And so we don't want to do that. So that's a fantastic question and a good segue into the next one. So that's talking about case switching, but it definitely applies here. So we don't want to cover it. Technically, it has to be monitored. How long do you think it should be opened? Let's say our case is delayed 5 hours. Do you think it's okay to leave it open for 5 hours? Some they're gonna pay you, you know, 20-something bucks an hour, 30 bucks an hour to sit in there and babysit the room for 5. What do you guys think?
I would say just break it down and then re-put everything back up when it gets closer to the surgery.
Yeah, and that's probably what would happen. If it's going to be like an hour delay, they will probably keep it up. But if it's going to be too much longer than that, they're probably going to break it down. And chances are, if they have another case, they're going to move a different case into there. If there's a 5-hour delay, if somebody can move up and into that spot, they're going to do that anyways, because they want to keep things going, right? So, you know, it should be monitored the whole time it's in there. What do facilities do a lot of times? They take a piece of tape, which is magic. Take a magic piece of tape and they put it across the whole door from jamb to jamb. And they write, "Do not enter," which flies know, like, not to go in there. So it's cool. Okay? Are you picking up my sarcasm? Can't see your faces, so.
So I have a quick story, like in regards to covering. So sorry, boys, this one you won't like much, but when my husband went in, my ex-husband went in, had his vasectomy done, I had been his MA. I was recovering, I was postpartum, and my husband went in to get his vasectomy and he had a new gal. And so she set up his vasectomy first thing that morning. She covered it in a drape and we walked in and I thought, well, geez, I wonder how long this has been here. And because we were like an afternoon appointment, and you know, he pulled the drape off and everything, did the procedure, and that man ended up with the worst infection in his testicles. It was unfreaking believable. You would, oh my. So yeah, that's how I know that you shouldn't cover stuff, because of the cooties. Yeah.
That's a hard way to learn that.
Right? He was less happy about it.
Yes.
But in hindsight, it turns out it's karma that got him first, but whatever.
They say it's, well, they say, you know, it's something. But yeah. So we had an issue when I worked at the Casa Grande Hospital before it was Banner for years and years. And we didn't have C-section room in labor and delivery. We did C-sections in the back. We did them in the OR. So eventually they did build a C-section room in the L&D department. And we staffed it for quite a while. The OR still staffed it until they could get staff trained. And what they ended up doing was taking labor and delivery nurses and labor and delivery techs, and they trained them to do C-sections. So one morning I go over there to see if the room needs stocked. And I walk into the OR and it's completely open and set up for a C-section. And I'm like, oh, so I went to the front desk and I said, it looks like you guys are getting ready to do a C-section, so I'll come back later to stock that room. And she's like, no, the C-section's not, it was like 3 hours from then. And I'm like, so how long has that been set up? And I'm like, you can't do that. Like, but you know, they really didn't know, because they've just kind of trained them up to do C-sections. And so that was a battle that we had to fight with them, because they didn't really have an understanding of all of these concepts like you guys will. And labor and delivery is a different animal. Like when I worked in labor and delivery, like the C-section is a 4-count thing. And the nurse is like, oh no, we don't do that here. We don't do that here. And we don't do that here. The techs would scrub in, lay everything out, go have some coffee, come back, literally stand, like not scrubbed in or anything, and like point at their stuff with the nurse to do their count. It was like bizarre. It's like weird. So you're gonna have to stick to your guns. For you ladies that wanna go to labor and delivery, stick to your guns, I'm just saying. Because a lot of L&D techs and nurses are just trained on the job, and they're not certified surge techs. They're not AORN certified as far as nursing goes.
I'll do it.
Yes, I know you will. I know all of you guys will. You guys are gonna set the standard. I know that. As far as covering something, we can't do that. So this kind of segues, like I said, into our next principle, where let's say we have a case that's scheduled in room 4, and for whatever reason, they're moving it to room 5 because room 4 is the neuro room and there wasn't a neuro case, but now there is, and they want to put it in there. Well, you've already opened and set up, or whatever the case may be. You can't cover it, wheel it into the other OR, uncover it, and call it good. Okay, you have to break it down completely, go get new stuff, go into the new room that you're assigned to, and open everything. Okay. And then the last one that we're going to talk about today is the STSR. So that's the surge tech in the sterile role. That's what that stands for, if you're wondering. This doesn't come from our book. This comes from another surge tech book that I have. But so us, the ST, we need to check the chemical indicators. We had a little bit of conversation about that already, to ensure the instruments have been exposed to the sterilization process. So if there isn't a chemical indicator, or I mean, if the chemical indicator has not been, hasn't changed, then we don't want to use it, right? And there's supposed to be an internal as well as an external chemical indicator for every single thing that we use. So both needs to be checked. But if the outside one is changed, but the inside one is not, what do you guys think? Is that okay? Or what's your opinion?
Not okay.
Yeah, it's not okay. The outside one really doesn't tell us much except for, yes, it went through a process, right? But it doesn't give us any indicator if the inside stuff is good. Let me see what Ian is saying. If any has failed, it all fails. Same thing as if there isn't one. Like there's an indicator on the outside, whatever that might be, and on the inside, there's no indicator whatsoever. We still don't want to use that, right? Because there's an absence of an indicator. So we would just want to get a new tray or a new whatever it is. Okay? Questions about these 3 little bits of principle 1? Okay, I have a couple videos for us to watch, and then we still have Kahoot, and then I'll let you go to take your post test, okay? What did I say? Oh yeah, videos. Okay, I haven't completely lost it. I've just almost lost it. All right, let's see, here we go.
And are we still taking notes on these videos as part of our lab?
Yeah, the videos are part of the lab, so if you have any takeaways, you can jot them down, but you don't have to take notes on every single thing. Just, you know, at the end, I would like there to just be some takeaways or some ahas, and so you can just jot down some new stuff if you would like.
Thank you.
Yeah. Oh. Go away, little strip of... Okay, here we go. This is this little tech tips guy. I like him.
Hey guys, welcome back to another Surgical Tech Tips. We're here still at CSN making some videos, and this is going to be going over opening up items onto a sterile field. Proper technique on opening up sterile items onto a field. Before we start opening anything up, I want to just break down the actual package itself. Some important things to note are these sealed edges around the wrapper of the peel pack are considered non-sterile when you're opening up the package. As you're opening up the package as well, the edges of the wrapper are initially considered contaminated around 2 inches of diameter. Now, as we can see here, these are our sealed portions of the package, kind of in a square, comes to a point. I like to start with just opening up the tip of the package until I get down to the straight lines or sides of the package. I will position the item so it's faced toward the field, and I will pull my left hand down and right arm forward to pop the package onto the field. Before you all start hating on me, yes, I know I'm supposed to be 12 to 18 inches away from the field when you're opening up a package. So when it comes to drapes or anything that's going to be wrapped, you will have kind of like a little tongue pointed toward you, the non-sterile person, as you're opening it up. You will pull that first and away. You have 2 more flaps inside that you will pull to the sides. And 1 more flap pulled toward you, the non-sterile person. And yeah, exactly. You check underneath and just toss it onto the field, just like that. An important tip that I always use as a student coming in here and opening up a field, if you don't have like a great basic pack that has all the items you need, and you're going to be opening up a lot of items that like to roll around on the backfield and potentially roll off of the field, like this S-mark bandage. Obviously it's in the shape of a circle, so it's going to roll as soon as it hits that table. So a lot of the times I will take items like this drape and specifically toss it onto the back of the field so I can use it as a backboard when I'm opening up items that like to roll. Now we've kind of gone over packs and wrapped items that we can open up onto a sterile field, but I really wanted to touch on items that are difficult to open up onto a sterile field. One being something like this, which is a clip applier. It's a plastic device, and as you can see, it's not just gonna fall out of here. The way this device and this company manufactures this is to really keep it in place by the plastic. So there's really no other way but to get a little bit closer to the field. I know it's against everything that your school and book says, but it's literally the only way. Another way of getting around this, if you only have like 1 or 2 items that are difficult to open like this, you can just keep it off of the field, scrub in, and have your nurse open it to you so you can just grab it right out of the package. You don't always have that luxury though, so I'm telling you right now, it's not always 100% what the book says. So that's about it. Very, very short little video on opening up items onto the sterile.
Okay, one more, and then we'll have a little discussion. So hold those thoughts. Let me do this. This is a dental office. This gal is talking more about dental stuff, but it applies to us as well. And this is about biological indicators. So chemical indicators, biological indicators, those are the things that we're going to put inside items. So as to give us an indicator that they're sterile or they've been through the sterilization process. Typically those peel packs have like an indicator like on the outside periphery, and then we put an indicator on the inside, or same with the tray. An indicator is going to go on the inside, we'll lock the lid on, we'll put indicators on the outside. And so there is one where she talks more about that, but since we're talking about microbiology, I thought it was more applicable to talk about biological monitoring. And these little biological monitors are for steam sterilization, and they have a live spore inside of them, a living creature inside of them called Bacillus stereothermophilus and Geobacillus stereothermophilus. And these bacteria make a spore. And a spore is like this extra protective armor that this bacteria can make, right? I think we've talked about those before. And then to top it off, to make it even more difficult for them to kill, they also love it really hot. All right, so we've chosen a spore for the biological indicator that is really hard to kill, so that it's gonna give us a good indicator that that stuff is sterile. And in the operating room setting, every single load that we put in a sterilizer, we're going to put in biological indicator. So just kind of, you know, setting up the stage. Let's let her talk about that.
I'm Leanne Kiefer. I'm a dental hygienist and director of clinical education for Crosstex International. As a dental professional, I have one question to ask you. What day do you confirm? Biological monitoring is the gold standard that's used to monitor the sterilization process. The CDC recommendation is that we are using biological monitoring at least once a week in our dental practices. So what I want to review with this session is appropriate placement of an in-office biological monitor into the sterilizer. The important thing to remember first is that you need to use two biological monitoring tests with the in-office. One of the vials is going to actually be placed into the sterilizer as your test. The other one is going to be your control. So this one will not be placed into the sterilizer. It's important that you write certain information so that you can identify the brand of the sterilizer, the run of the day, the date, and the operator's initials. When writing on the vial, it's recommended that you use an approved marker that can be processed within a steam sterilizer. When you have the biological monitoring vial, I want you to note that there is a strip, a colored strip on the outside. This is actually a chemical indicator that's used as a process indicator, because at the end of the sterilization cycle, that blue line should turn black, which means that this vial has already been processed. You confirm in-office biological monitor can only be used in a steam sterilization cycle. This particular confirmed product cannot be used in dry heat, cannot be used in a chemiclave cycle. You have a choice. You can either place the vial directly into the sterilizer on the tray, or if you want to assimilate what you're doing with your instruments, you can also place it into a sterilization pouch. When you place the vial, you want to place it in the most challenging part of the sterilizer. That means the place that you're most likely to have an air pocket. That area is going to be on the bottom tray of the sterilizer. It's either going to be over the drain, which may be in the middle of some autoclaves. In other autoclaves, it's in the very back left-hand corner. The other place would be to position it by the door where there may be an air leak if the gasket doesn't seal correctly. Again, you'll note that we're not placing it over any of the instruments. We're placing it in a single layer. We're going to place it paper side up because this is a gravity displacement sterilizer. At the end of the cycle, it's important that we wait 10 minutes because the liquid inside that vial is 121 degrees Celsius. After you've waited the 10 minutes, you'll take the test out of the steam sterilizer, and the first thing that you're going to look at, did that blue chemical indicator strip turn black? You will then add the control vial from the same box, the same lot number, and you will process them both in the incubator depending upon if you have a 10-hour or a 24-hour product, which we'll talk about in our next video.
Excellent. We're not going to watch that, but appreciate it. Okay, so when you guys go out to your clinicals, typically what will happen is your first week or so, some sites don't care at all. If you go to sterile processing, other sites require you to be there for 2 weeks, which I think is too long because you're surge techs, not sterile processing techs. But suffice it to say, you will spend some time in sterile processing. And that will help you learn kind of the other half of the picture, right, of what happens in the process. So you might be checking those biological indicators. You might be loading them in the incubators. You might be filling out the paperwork regarding those biological indicators. So what the video doesn't say is once they've been in the incubator for however long they're supposed to be and you take them out, the one that is the live spore is should have changed color, your control, the one that you didn't cook, because you're gonna, the top has like you know how a glow stick has like a little thing that you crack to activate it? The biological has the same thing, but it's a little lid. So you push it down, and what happens is there's food in there, spore food, and they think it's really delicious. And so in the control, we should have live spores. They're gonna eat that food, and they're gonna put off some toxins, and it's gonna change the color of that fluid. The other one shouldn't change at all if the spores are all dead, right? So that is our confirmation that our stuff is sterile. And we should not put that sterile stuff out into circulation before that biological has been read. Does it happen? Yeah, sometimes it happens, right? You have a vendor that brings in a tray late and you don't have, I think the incubators, it's faster now. But sometimes you have to get that tray straight to the OR and they don't read the biological first. But it should be read first because it's our confirmation that if all the spores are dead, then we're probably good. Now, different from the one she showed in the video, in the operating room setting, we're going to use, it's like a pack. And it has a whole bunch of cards and the biological is in the center of that. So it's even harder for the steam to penetrate that thickness of that package. So we're giving even more resistance than the normal resistance that comes with it being a spore and it being thermophilic, right? Loving the heat. All right, and we want to make it as difficult as possible because that makes us more sure we can sleep at night that the stuff that we're sending to the OR is sterile. Okay, that's all I got. What do you guys got? How was it today? It was long today. It won't be as long because we want like to talk about like class overview.
Do we still have the time to do the Kahoot and the post-test?
Yeah, we're going to do the Kahoot right now. It's about 20 questions, so it shouldn't take us too long. And then if you guys need extra time, I can always change the settings for the post-test.
Okay.
Let's see, I was just going to see what Lexi was saying really quickly. Well, yeah, probably, Lexi, because the chance that you're going to go to the sterile processing department where you're doing your clinicals, I mean, is that where you're going to go? I don't know. So probably you're going to have to. Yeah, that would be my guess. All right. Yeah, you're welcome, of course. Okay, so we're going to jump to the Kahoot. Let me see what Gabe says. So again, if you go to your clinical site where you work, that might be a different story. But if not, they're not going to take that. And well, you know, I mean, I don't know. Rulan's a sterile processing tech. So if that's the case, maybe, since that's already your job. Otherwise, you know, some sites are pretty persnickety about putting you in sterile processing at their place. So you learn how they do it and how their trays are and what their processes are. So, you know, different strokes for different folks depending on what facility you go to. So I guess we'd have to just kind of play that by ear. Yeah. Okay. Yeah, you're welcome. I mean, if we can get you out of it and get you straight to the OR, let's do that. But it is good to see what your site is doing as far as it helps you learn what's in a major tray, what's in a minor tray, what's in a ortho tray, you know, because you're putting them together. So then when you get to the point of being in the OR, you have a better understanding of what their trays have and, you know, those kinds of things too. So, but I can see both sides of that. I don't mind sterile processing. I just don't like decon. I don't like that washing dishes part. No. Okay. Let's see. Let me get Kahoot going here. It's right over here. Make it quick.